So proteins are some of the most abundant and diverse macromolecules that are found in living organisms, and they perform a variety of functions in the human body. And I have listed some of those functions below. So proteins can act as enzymes, which are biological catalysts that help speed up chemical reactions in the human body. Proteins are also hormones. So you have your insulin, progesterone, testosterone, and estrogen. So insulin is an example of a protein hormone, which acts as a chemical messenger. So it sends messages and signals between cells and across the body. You also have transport proteins such as hemoglobin that transports carbon dioxide and oxygen throughout the body between different cells. You also have proteins that act as antibodies or immunoglobulins, which are produced by white blood cells, and they act as a defense mechanism against our body and guard us from getting infections and diseases. And we also have structural proteins, and these are the ones that provide structure and stability to different parts of the body. Two examples of structural proteins are keratin, which is found in our nails and in our hair, and collagen, which is connective tissue. So, like many other organic molecules, proteins is what we call a polymer. So polymer, again, is many, many small little units called monomers joined together to form something massive or big that's called a polymer. So just like any other polymer, proteins are also made of a basic subunit, or what we call a monomer. And that monomer is what we call an amino acid, and this is what an amino acid looks like. So this part, we have a central carbon, which is what we call the alpha carbon. And on this side of that, on the left side of that carbon, we have the NH2 bit, which is your amino bit. And above the central carbon, we have a hydrogen atom. On the right side of the carbon, we have a carboxylic acid group, hence we have the acid, bit of amino acid. And we have a bit here called R, which is different in every single amino acid. And the chemical nature of the R subgroup is what will determine the chemical properties, functions of these amino acids, and how they bond to form major proteins and polypeptides, which I will go through later on. So say, for example, we have a simple amino acid called alanine, which looks a little bit like this, where the R group is just CH3. So that pretty much makes it a nonpolar and uncharged amino acid. So basically this amino acid will tend to form more hydrophobic interactions as opposed to more hydrophilic ones or hydrogen bonds between in polypeptide chains. So that's your alanine. You can also have something like aspartame, which is a little bit different. So aspartame looks a little bit like this. So the fact that there is a negative charge on the C double bond O part of aspartame gives the whole amino acid a negative charge, which makes it more polar and makes it more prone to interacting in polypeptide chains using hydrogen bonds and more polar bonds as such. You can also have something like this amino acid, which CH2, CH2, and you have NH3+. And this one has an overall positive charge. And again, so this is a positive polar charged amino acid. So depending on what the R group looks like, the charge of the amino acid is determined, and that subsequently will determine how it bonds with other amino acids and also the function of the overall protein it bonds. Now in our body we have 20 amino acids, and out of these ones 9 are essential amino acids, so those are the ones we can only get in our body from our diet, and we also have 11 non-essential amino acids. So far we have looked at the structure of a protein on a more microscopic or a very basic level. Now let's look at the more big picture structure of what a protein looks like on a macroscopic level, and that is essentially divided chronologically into 4 sequential groups of complexity. So you have your primary structure, which is the most basic one, and then you have a secondary structure of protein which gets a little bit more complex, the tertiary structure even more complex, and quaternary structure, which is the highest level of complexity in the structure of a protein. Now, the primary structure is essentially the sequence of amino acids. So AA I'm drawing here is your amino acids that are joined together by what's called a joined together by what's called a peptide bond. So say for example I have alanine, cysteine, and aspartame. We've got 3 amino acids here, and they are joined together by these two bonds. And this one is what we call a peptide bond. So that's the bond between two subsequent or sequential amino acids. And these peptide bonds together, they end up joining many, many amino acids together to ultimately form what's called a polypeptide. So like we looked at polymer, polypeptide is many, many peptide bonds that are created by the joining of many, many amino acids put together. That represents the primary structure of a protein. And the primary structure of the protein is crucial in determining its function and also how its secondary, tertiary, and quaternary structure look like. Now, the secondary structure of a protein is a little bit more complex. Now, that is created by how the polypeptide chain starts to fold amongst itself, essentially. And that can give rise to two shapes. So you have one shape that looks a little bit like this, like your old telephone cord, or like your DNA double helix, and true to its name, it's called alpha helix. And you can have another shape of the secondary structure of an amino acid that looks a little bit like this. It's what we call beta pleated. And how this folding takes place is a little bit like this. So if we go back to our structure of amino acids, we have the NH group and the COOH group that's over there. So what normally happens is the NH group, say over here, we have that over here, and your C double bond O group over here. What they will do is they will come together and form a hydrogen bond. So if I do that, it's called the hydrogen bond. And that hydrogen bond between them is what actually causes the polypeptide chain to coil up or fold a little bit. And you have the same story in the beta pleated one as well, in that you actually form the C double bond O and NH, same story, but that bonding occurs this way, across what we call pleats. But it's the same hydrogen bonding principle, and they end up forming the folding of the polypeptide chain gives rise to the two forms of secondary structure of a protein, which is the alpha helix and the beta pleated. Now the tertiary structure is the next level of protein structure, which is the overall three-dimensional shape of the protein. So the tertiary structure is what gives the protein its overall three-dimensional shape, appearance, and therefore its functions. And that is created by, it could be hydrogen bonds, it could be disulfide bonds, it could be hydrophobic interactions between the R groups of subsequent amino acids. So it's the same thing, except unlike secondary structure where you had the C double bond O and NH forming hydrogen bonds, you have hydrogen bonds, hydrophobic structures, and disulfide bonds, various different bonds that are formed by the R group of adjoining amino acids. And this, as I said, is mainly responsible for the function of the protein. And the last level of the protein structure is what we call the quaternary structure. And that is formed by the polypeptide units. So you've got your different polypeptide chains. They form what's called subunits that give the protein either a globular appearance or a fibrous appearance. And obviously, depending on whether it's globular or fibrous, its properties and its function differ. So a globular protein is something like your insulin or your hemoglobulin, hence the name globulin over there. So globular proteins overall, they look a little bit like, they've got a bit of a round shape, whereas a fibrous protein is a little bit messy like this. And we find that in, say, something like collagen or, say, keratin, or even the protein that forms silk. So these are the different structures of proteins that we have looked at. Now, proteins are quite a popular exam question or an exam topic. And I have used some examples from the International Baccalaureate exam board. And we're going to look at two types of questions that you commonly get asked and how you can actually answer them and get the marks that are required for it. So we're just going to look at some exam strategies now. Okay. So a typical short answer question can be, draw a labeled diagram of the structure of an amino acid. So this typically will be a 3-mark question. So before you attempt this question, I always emphasize on highlighting what the key words are in that question. So, first of all, look at what your action verb is. So it's asking you to draw. So you don't have to write a long story, you don't have to give any descriptions, you just have to draw something. But you can't just draw something and not tell the person who's marking your exam what exactly it is. That's why it's important that your diagram, whichever diagram you draw, that even if it doesn't say, the question doesn't say it has to be labeled, by default assume that all your diagrams have to be labeled. So in this case, because it's giving you that instruction, you have to label your diagram, otherwise you lose your marks. You have to draw a label diagram of the structure of an amino acid. And this is all you have to do. You show your amino, you draw a little bit like this is a structure that looks like this. You've got your central carbon, your alpha carbon, you've got your amino bit, you've got your acid bit, you show your R and you show your H. But it's very important that you label which is which. So, you show your amino bit and label it correctly, you get 1 mark. You show your acid bit and you label that correctly, you get 1 mark. You get half a mark for drawing your hydrogen atom correctly, and you get half a mark for showing your R group. And this can be the other way around as well. It doesn't matter as long as it's there and as long as you're showing it. And by doing this, you get 3 marks, nice and easy, very simple. You don't have to give any descriptions, you don't have to say anything else. So these are 3 very simple, straightforward way of getting 3 marks for a short answer, a typical short answer question. Now, there's another typical question that you can get that's more that requires more of a lengthy response. And that can be something like, describe the structure of proteins. Now, these ones tend to be more 8 marks or 9 mark questions, and people normally look at it and go, they get a little bit overwhelmed because they're not really sure how much to include, how little to include, what not to include, and also how much time they should spend on a question like this. And essentially what you need to do is, again, what I showed over here, is you identify what the action verb is, You got describe, so you can't just label something like this and leave it. You actually have to give a little bit more detail. You have to make it more descriptive. You have to tell a little bit about what it looks like. You can draw diagrams. I would highly recommend drawing diagrams just to supplement your text or whatever you write about it. That will really show a good structure and a good understanding to the examiner that you know what you're talking about. And it's, again, the structure of a proteins. So you can't just leave it at, use one example. You have to describe the whole thing of what a protein looks like. Now, simple way of doing that is you, first of all, you write down the fact that you have your primary structure, you've got your secondary structure, tertiary structure, and quaternary structure. So the fact that you just mentioned that alone, that can get you 4 marks. So 1 mark for each different type of protein structure. And just mention 1 or 2 sentences about each type of the definition or the description of what each type of protein structure is. So say, for example, when it comes to the primary structure, you just say that it is the sequence of amino acids that are joined together by peptide bonds to form a polypeptide. So when you say all these things, you get an extra mark. And again, to supplement that, you can draw a diagram just to supplement what you're writing. Similarly, when you get to secondary structure, you can do the same thing. That you have to mention the fact that the polypeptide chain, there's folding in the polypeptide chain that gives rise to these 2 shapes, which is your alpha helix and your beta pleated, and those are joined together by hydrogen bonds. And again, you can draw a diagram just to be a little bit more descriptive and supplement your text. You have your tertiary shape, which again, you have to mention that it's a 3-dimensional shape. And by doing this, you get 1 mark. You have to mention that it's a 3-dimensional shape. And that is again is caused by further folding of the polypeptide chains from the secondary structure. And you have to mention the fact that, you can mention the fact that it's hydrogen or bonds between the R groups of amino acids. So by mentioning that, you can get another mark. Got 1 more mark. And a quaternary structure is when you have your polypeptide chains bonding further to form a subunit or subunits that give it either a globular or a fibrous appearance. And that will get you another mark. Draw diagrams, but I would also highly recommend, if you're getting a little bit overwhelmed about how you structure this answer, you can do it in 2 ways. You can do it either through dot points like this. You can say primary, secondary, tertiary, quaternary, and add your 1 or 2 sentence descriptions as dot points. You can add them in proper sentences as well. That's absolutely fine. What you can also do just so that you're not getting overwhelmed, is you can draw or you can organize your information in a table like this. So what you can do is, what you can say over here is protein structure. That's one column. You can also have, then you've got your description, and you have your diagram. Sorry, this is not very clear, but hopefully you'll get a picture of what I'm trying to say. So say, for example, the first column will be primary. You add the description of the primary, and then you draw a diagram showing that. Then you can have secondary, draw that, and you do the same thing for tertiary and quaternary. So whether you do it this way or that way, whatever helps you easily organize and arrange your information, it should be able to get you the marks that you want.